Hello and welcome back to your Chem 4 lecture. Today we are continuing into the lipids chapter, chapter 15, by talking about waxes and triacylglycerols, two closely related classes of compound that, we'll explore here in the lecture today. Now, waxes, we're going to start with waxes today. It's a class of compounds where you have basically an ester with a saturated fatty acid on one half, with a long chain alcohol on the other half. So both of them are each going to contain 14 to 30 carbon atoms on their own. So like you'd have a 14 carbon chain fatty acid esterified with a 14 carbon chain alcohol. Now, what are something that we notice about this? Like that's pretty big numbers, right? That's a lot of carbon atoms for both sides. And the more carbon atoms we have, the more carbon-carbon bonds, the more carbon-hydrogen bonds that we have, the less polar it's going to be. And in fact, waxes are very nonpolar. You can see that because, for example, when you look at leaves, the leaves of plants are coated with a type of wax. And that wax basically prevents water from just seeping into the leaf. And that's why you see water bead up on these nonpolar waxy surfaces. So you can look at a few different types of waxes here. So for example, beeswax has a 30 carbon alcohol chain esterified to a 16 carbon fatty acid. And this is present in honeycomb. And now you can use this for a lot of different things like honeycomb candle or beeswax candles are fairly popular. Beeswax can also be kind of a sustainable way of making shoe polish or wax paper. Carnauba wax, which you could use for your like surfboard, for example. You have 26 carbon fatty acid chain and a 30 carbon alcohol. And jojoba wax is another example here, popular in candles, soaps, and cosmetics, with a 20 carbon alcohol and a 20 carbon fatty acid. So just giving you an idea here, basically you take two long carbon chains, one carboxylic acid, the other an alcohol. When you put them together in an ester form, that gives you this wax. Okay, now waxes are interesting, but as it turns out, like the most important type of lipid for our cells are triacylglycerols. So fatty acids, there's a lot of them, and your body needs to store a lot of them as a way to store energy. And the most convenient way for the body to do this is to store them as what's called a triacylglycerol. We're going to look at this on the next slide, but what a triacylglycerol is, it's basically like a triple ester. You take glycerol, which is a trihydroxy alcohol. I can draw it for you real quick here just to make sure that we're on the same page. Basically, it's carbon, 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 so 3 carbons. And then you put a hydroxyl group on each of them. So OH here, OH here, and OH here. And each of these hydroxyl groups is going to be involved in an ester with a fatty acid. Hence the word triacylglycerol can stick 3 fatty acids on this one glycerol molecule. So the way that we actually name it is you take glycerol as a molecule, you're going to name it glycerol, and then name the fatty acids as the carboxylate salts. So take the ic acid ending and change it to an ATE ending. So for example, glycerol here, this is another way to draw it. Like this is the condensed structural formula. You can form a triple ester or a triester with 3 stearic acid molecules. This happens in our body all the time. And what happens is you form an ester bond 3 separate times. So this carboxylic acid and this alcohol becomes an ester, this carboxylic acid and this alcohol becomes an ester, and this carboxylic acid and this alcohol becomes an ester. So the triacylglycerol that's formed is something that looks like this. You have, in this case, glycerol as the first name, and then you have 3 stearic acids, which became stearate, so you call it glycerol tristearate. The common name is tristearate, but I'm not going to require you to memorize that. Now, you don't just happen to have all 3 of the same fatty acids. You can also have combinations of different ones. So like a triacylglycerol here has a stearic acid, an oleic acid, and a palmitic acid. And we just want to point out that this is a mixed triacylglycerol, which is it's one of the possibilities of triacylglycerols. Okay, now why are triacylglycerols so important? Well, they're the major form of energy storage for animals in particular. So, for example, animals who hibernate will eat a large quantity of plants and other nutrients that are high in calories. And basically, the energy in those food sources will be stored in the form of triacylglycerols. So hibernation, you have to eat a bunch of that stuff, and then as the outside temperature drops, hibernation is just like a low metabolism, low cellular activity, low respiration, low heart rate state. Now, we don't do that, but for example, bears do that. Polar bears are able to do that, and other types of bears. During that time, they're not consuming any sugars, so the normal glucose cycle that we're going to talk about in a future chapter is not able to be used. The only thing that's left is the triacylglycerols that store that. Okay, so this is inconvenient here because you're going to need to refer to the notes in the previous lecture. But I'm asking you here, what fatty acids make up the following triacylglycerol? Again, you'll check your notes and you can see what the names of these fatty acids are. Note here, this is saturated, and this is saturated, and then this is clearly unsaturated. So refer back to your notes of the types of fatty acids that we talked about. It's one of the ones that we covered in the previous lecture, and then identify which ones they are right here. So pause the video, go back to your notes, and when you come back, we'll take a look at the answer. So pause. Okay, let's take a look. So here, up here, we have 16, 17, 18 total carbons in an unsaturated fatty acid. There's no double bonds here. So that's basically an 18-0 fatty acid, which is stearic acid. Down here, another easy one, it's 12, 13, 14 carbons, no double bonds. So that's 14-0 unsaturated. That's myristic acid. Trickier is here in the middle. We have, so 1 plus 7 is 8, 9, 10, plus another 8, 18 carbons. And we have a double bond in the middle. This represents oleic acid, one of our monounsaturated fatty acids. Okay, so now let's go ahead and talk about how we draw the structure of a triacylglycerol. Let's say that we have a triacylglycerol named glycerol tripalmitolate or tripalmitolene, and we want to draw the structure for it. Well, it's pretty simple to draw it because what we're going to do is here. Let me go back over here, because the way you can think about it is basically you have a cap, and this cap is the glycerol. Okay, so you start from here, and now each of them is going to be esterified to your fatty acid. So they're each going to have this oxygen bonded to a carbonyl, just like that. And the chain of the fatty acid, that's going to be determined by the whichever fatty acid is actually involved in the ester bond. So here it's tripalmitolate, so that means it's palmitoleic acid. So here. So if we check that out in our notes, we know that we've got, that's 8 carbons followed by a double bond, followed by another 6 carbons after. So you can start to try to draw it here. I'm drawing it in the line angle formula. You can also use the condensed structural formula here. But here we have carbon 1, 2, 3, 4, 5, 6, 7, 8. Okay, so that's, now we're up to here, right? This is carbon 8. So then carbon 9. Then we have a double bond. Okay, so we have a double bond like that. Now, we know that this is a cis double bond. This is going to be hard to draw, but we can still do this here, followed by another 6 carbons. So 1, 2, 3, 4, 5, 6. And then you would just do that 3 times. Okay, so why don't you go ahead and give me the condensed structural formula for the triacylglycerol that contains 3 molecules of myristic acid as the fatty acids. So pause the video here, work it out, and we'll take a look at the answer when you come back. So pause. Okay, let's take a look. So what happens here is, again, you have the same cap as before. It's a glycerol. That's 3 hydroxyl groups here. And those each become the alcohol side of 3 separate esters. And fortunately, myristic acid is a saturated fatty acid. So there's no double bonds that we have to worry about. We can just draw 14 carbons in a row. Okay, so now let's talk about the word fats and oils. A lot of times these are used interchangeably, but they do have some differences. So the difference between a fat and an oil is that a fat is usually a solid at room temperature, and fats tend to come from animal sources. So you can have things like meat has fat in it, as well as lard, which is processed animal fat. Milk, butter, cheese tend to have fats. Oils are usually liquid at room temperature, and they are usually obtained from a plant source. And now we know something of the difference between them, right? We know that fats animal fats tend to be saturated, and plant fats, or oils, excuse me, tend to be unsaturated. So, for example, oils that come from olive or peanut are the monounsaturated oils. They tend to have a lot of oleic acid. Corn, cottonseed, safflower, and sunflower seed oil are polyunsaturated. That means that they have 2 or more double bonds in the fatty acids. And then finally, palm and coconut oil are outliers. They're solids at room temperature because they have mostly saturated fatty acids, unlike the other plants that we've been talking about. Okay. So we've talked about it before as far as saturated fatty acids versus unsaturated fatty acids. Saturated fatty acids have higher melting points because they're able to pack together more tightly. And you can find the saturated fatty acids in animal fats, as well as coconut or palm oil as well. Vegetable oils tend to have very low melting points because if you look at it here, this is an example of a triacyl glycerol involving oleic acid here, which is again a plant fatty acid. Because the molecules don't pack together very well, they tend to have very low melting points, and they'll be liquid at room temperature. So that's kind of a usual thing that occurs with plant oils. And yeah, just pointing that out here again, because of the high percentage of unsaturated fatty acids in vegetable oils, they tend to be liquid at room temperature. Okay, well, let's go ahead and talk about this here. Okay, so first up, triacylglycerols can be hydrogenated. So remember, we talked about hydrogenation when we talked about alkenes a long time ago. So if you have an unsaturated fatty acid, you can use the same process that we talked about to hydrogenate alkenes with the fatty acid. And remember, what you do basically is you take you heat up the oil in this case, you add in a nickel catalyst or some other palladium, platinum catalyst works as well, and bubble in hydrogen gas. And what happens is the carbon-carbon double bond will be broken, and you'll form 2 new carbon-hydrogen bonds. This is a way of taking unsaturated fatty acid and producing a saturated fatty acid. So for example, if you take glycerol trioleate, remember that's the one that we looked up here. That's this one right here. Again, it's a liquid at room temperature. If you hydrogenate it, you actually get glycerol tristearate or tristearate. All of a sudden now you've got saturated fatty acids in your glycerol tristearate, in the glycerol tristearate, and that means that it's going to be a solid at room temperature. This is actually what's behind the synthesis of margarine, for example. Margarine is basically where you take a vegetable oil and you hydrogenate, forming a unsaturated, or going from an unsaturated set of fatty acids to a saturated set of fatty acids. And that gives you a solid vegetable oil that you can use, as like a substitute for butter. Okay, So let's say that we have glycerol trioleate, and we do a hydrogenation reaction. What's going to happen? Does it split into glycerol and 3 oleic acids? Does it form glycerol and tristearate, which is again a saturated triacylglycerol? Or does it form glycerol and 3 stearic acid molecules? Well, hydrogenation, as you'll remember, just takes the double bond and turns it into a single bond and adds the 2 extra hydrogens that are needed. So what's going to happen is your glycerol trioleate forms glycerol tristearate. It still stays as a triacylglycerol, just the identity of the fatty acids change. Okay, so I think I mentioned this previously in the last lecture, but one of the links to health that's important to note is that you may have heard of is this idea of trans fatty acids. So remember, unsaturated fatty acids in their natural form from plants tend to be cis. And so that means basically you have your carboxylic acid and a long carbon chain on the same side of a double bond. However, through processing, they can turn into trans fatty acids, where the long chain is on the opposite side as the carboxylic acid chain. So this looks more like a saturated fatty acid. So when you hydrogenate your vegetable oil and you form your saturated fatty acids, sometimes some of those double bonds start out cis and they end up as trans double bonds. So that changes the overall structure of the fatty acids. Now, the problem is trans fatty acids behave like saturated fatty acids, but they raise LDL cholesterol and lower HDL cholesterol. So they have a deleterious effect on your health. So what happens is basically you have an isomerization rather than the saturated product that you actually want. And these trans fatty acids tend to be can be real problematic for your again your cholesterol levels in your blood. So naturally occurring trans fatty acids come from milk, eggs, and beef, but any trans fatty acid, like from a hydrogenation process, can come from anywhere that oil is used. So deep fried foods tend to use saturated or hydrogenated vegetable oils. So anything that has a hydrogenated vegetable oil will have trans fatty acids in it. So that's everything from margarine that we talked about already, vegetable shortening like Crisco, for example, and then commercially produced crackers, chips, all of these things tend to have hydrogenated vegetable oils in them, and they will contain trans fatty acids. Okay, so let's go ahead and go through here. Which of the following statements are true and which are false? A, there are more unsaturated fats in vegetable oils. B, vegetable oils have higher melting points than fats. C, hydrogenation of oils converts some cis bonds to trans double bonds. And D, animal fats have more saturated fats. Go ahead, pause the video here. Take a look through these statements, try to identify what's true and false, and we'll take a look at the answer when we come back. Okay, let's take a look here. So first off, are there more unsaturated fats in vegetable oils? Yes, that is true. Remember, unsaturated fats means the ones that contain double bonds. So the saturated tend to be animal fats, the unsaturated tend to be vegetable oils. Next up, vegetable oils have higher melting points than fats. That's clearly false. Fats are solid at room temperature. Oils are liquid at room temperature. The liquids must have a lower melting point. Hydrogenation of oils converts some cis double bonds to trans double bonds. That's what we were just talking about with trans fatty acids. That is true. And then finally, animal fats have more saturated fats. That's the obverse of A. So that is also true. Okay, so go ahead and draw the condensed structural formula for the product of glycerol tripalmitoleate after it undergoes hydrogenation. So we're going to do complete hydrogenation for glycerol tripalmitoleate and show me what that's going to look like. And you can do the condensed structural formula. You can also do the line angle drawing if you prefer. So go ahead, pause the video here, come up with those products, and then we'll take a look at the answer when we get back. Okay, so the main thing that's going to happen here through hydrogenation is each of these double bonds is going to be converted into a single bond. So whereas before we had, 8 carbons, then a double bond, and then 6 carbons. Now it's just going to be 8, 10, 16 carbons all in a row. So you have your ester carbon followed by 14 CH2s and then capped off with the methyl group. And that's just 3 separate times. So remember, hydrogenation, that's just where you're going to take a double bond and turn it into a single bond, filling the carbons with hydrogens. Okay, now you may notice that these triacylglycerols are esters, right? We've talked about that multiple times. They have these ester bonds. Well, if they're esters, that means they can do the same chemical reactions that esters can do. So triacylglycerols, for example, can undergo hydrolysis. And in the case of hydrolysis, what happens is the glycerol is going to be separated from the fatty acids. Like this is glycerol tripalmitate. So you have 3 palmitic acids esterified into 1 glycerol. Using using an acid catalyst or a particular type of enzyme called a lipase. We'll talk more about those in the next chapter. You can actually split this apart into the glycerol half and the 3 palmitic acid molecules. So that is acid hydrolysis. Now, if you recall from our ester chapter, we had acid hydrolysis and we had base hydrolysis, also known as saponification. And saponification literally means soap making. So if you do a saponification reaction with a triacylglycerol, you literally form a soap, which is anything that is a salt of a fatty acid. So sodium hydroxide, for example, forms the sodium fatty acid carboxylate salt. And with sodium as the counter ion, that gives a solid soap. Potassium hydroxide gives a softer or more liquid soap. So for example, saponification of glycerol tripalmitate here, you form the glycerol molecule just like you did with the saponification of esters. And this time you form the carboxylate salts of the fatty acids. So this is sodium palmitate in here, for example. And that's just one example of the top types of fatty acid carboxylate salts that you can form. Okay, so here's your summary table for your organic and lipid reactions. Remember, you have the esterification, which is just the same as normal. It's carboxylic acid plus alcohol gives an ester and water. In our biochemical processes, that's where we take 3 fatty acids, react it with glycerol. And again, an enzyme actually does the reaction, but you could use triacylglycerol. Hydrogenation also occurs, where before we talked about alkenes being hydrogenated to form alkanes. Now we have unsaturated fats being hydrogenated to form saturated fats. Hydrolysis would be where you take ester with water in a acid catalyst, and you form your carboxylic acid alcohol, so you split up the ester. The same thing happens with lipids. It's your triacylglycerol this time, and it's an enzyme doing the catalysis to form 3 fatty acids and glycerol as your product. Saponification we just talked about. Again, that's where you take your triacylglycerol, 3 equivalents of your base, like sodium hydroxide, and that gives you the salts of your fatty acids along with the glycerol. Okay, so go ahead and give me the products here for this equation. I want to see the products for the hydrolysis of glyceryl trilaurate or trilaurate, which is given to you here. And again, in this case, the catalyst is lipase. So think of this as the same as your acid catalyzed hydrolysis. Go ahead, pause the video, show me your answer, and then we'll take a look at the answer together. So go ahead, pause. Okay, so basically lipase tells you that you're going to have a hydrolysis of the triacylglycerol, where you're going to have glycerol as the cap molecule over here. That's your alcohol. And the fatty acids are going to be produced are the fatty acids that are involved in the triacylglycerol here. So this is basically just going to split it in half, give you 3 of these lauric acid molecules along with 1 glycerol. Okay, so that does triacylglycerols. Next up, we want to talk about phospholipids. And phospholipids have a particular structure to them. They're important because they're going to be involved in the next set of compounds that we want to talk about, which is we want to talk about cell membranes eventually. And so we're going to show kind of the way that it looks here with phospholipids first. Okay, so the way that phospholipids work, we're going to talk about two different ones. We've got glycerophospholipids that contain glycerol, and then we have sphingolipids that have a different structure. So glycerophospholipids are the most important ones, and that's what we want to start by talking about. The structure looks like this. You have your glycerol as a cap. Now that glycerol is going to be esterified twice to two fatty acids, usually the same fatty acid, but could be different fatty acids. Now, here's the difference. The third alcohol on glycerol is not going to be esterified to a carbon. Instead, it's going to be connected to a phosphate, an organic phosphate. So PO4 should look familiar. Remember, PO4 with a 3- charge is phosphate. In organic compounds and in biological chemistry, phosphate forms really important linkages. For example, in the backbone of our DNA, and also here in glycerophospholipids. The phosphorus takes the place, basically connecting the glycerol to an amino alcohol. Now, the word here is phosphoester, so it's similar to an ester bond, but it's distinct because it's got a phosphorus rather than a carbon. Now, one that's even weirder than that is sphingomyelin, for example. Sphingomyelin, rather than glycerol, has a more complicated molecule called sphingosine. Now, you don't need to know the structure of sphingosine. You just need to know that sphingomyelin contains this backbone sphingosine that's connected first to a fatty acid and then via a phosphate to another amino alcohol. So check out the differences here. Glycerophospholipids have a glycerol and two fatty acids. So it's very close to a triacylglycerol. But then the third one is bonded to a phosphate that's bonded to an amino alcohol. An amino alcohol just means it's both an amine and an alcohol, as both of those functional groups. Sphingosine, almost the same, but backbone here, sphingosine rather than glycerol, one fatty acid, and then a phosphate connected to an amino alcohol. Okay, so let's briefly mention the types of amino alcohols that can be found in the glycerophospholipids or in sphingomyelin. You have choline here, and choline is, you can see you have an alcohol hydroxyl group on this first carbon here. And then we end with a positively charged ammonium that's bonded to 4 different methyl groups. So again, it's an amino alcohol, that's choline. Serine is next. Now, serine is going to become important eventually because it's also an amino acid. But here, the amino alcohol form is we have a hydroxyl group on this end, and then an ammonium up here, 2 carbons away. Then finally, the simplest amino alcohol, which is ethanolamine, which has your hydroxyl group, then 2 carbons, then an amine group. Now we have it as the ammonium ion here because at a physiological pH, these will be ionized at that pH. Okay, so let's talk about 2 different types of glycerophospholipids that are really important for brain and nerve tissues. We've got lecithin and cephalin. So lecithin, for example, is first off, it's a glycerophospholipid. So again, that means you have glycerol, 2 nonpolar fatty acids, and then down here, we have a phosphate that's connecting to, in particular, choline. So lecithin contains choline. Cephalin, on the other hand, contains ethanolamine. So again, the idea here is the fatty acids, glycerol, almost all the same. The only thing that's going to differ here is the amino alcohol. So the point or the good thing about glycerophospholipids is that they have a polar region and a nonpolar region. Like, for example, looking here, we have a positively charged nitrogen, and then we have this phosphate that retains a negative charge as well. So this is a highly polarized phosphate amino alcohol combination here. Now compare that to our nonpolar fatty acids. So we have a polar head up here and a nonpolar tail down here. So the nonpolar tail will only dissolve in nonpolar substances like lipids, and then the polar head that contains the phosphate and the amino alcohol is going to be strongly attached to water. So this is actually what gives cell membranes their structure. You have a polar outside, and then inside, the nonpolar tails are all interacting with one another. So yeah, here's another example of your glycerol phospholipid. You have your nonpolar tail and then the polar head. And this is going to become really important when we talk about the cell membrane structure. Okay, all right, here we go. So sample exercise number 11, go ahead and tell me, given what we've learned so far, identify each of these as either fatty acid, a triacylglycerol, an amino alcohol, or a glycerol phospholipid. We have four things to talk about here: glycerol triolate, cephalin, choline, and palmitic acid. So pause the video, look through your notes, and identify each of these as one of the four categories here. Okay, so let's go ahead and take a look. So glycerol triolate is clearly a triacylglycerol, right? We see this word glycerol in the beginning. That's a giveaway that it's a triacylglycerol. Cephalin, remember we had cephalin and lecithin as the two glycerophospholipids. Choline is one of the amino alcohols, and then palmitic acid is a fatty acid. Okay, so now given all of that, can you draw me the condensed structural formula of cephalin, acephalin, that contains 2 stearic acids, and you don't need to memorize it. There's 18 carbons in the fatty acids of stearic acids and zero pi bonds. So there's no alkenes to worry about. And serine, of course, in the ionized form as again, a glycerophospholipid cephalin. So if you check it back out up here, remember, this is our general look at lecithin and cephalin. Go ahead, look through your notes, and draw the condensed structural formula for cephalin that looks like what we just saw in the lecture notes right now. So pause the video, and when you come back, we'll have the answer. Pause. Okay, so the key here is you always want to kind of do the same method of drawing as what we were seeing back up here. So for example, you just want to start by drawing the glycerol as the backbone. So CH2, CH, CH2. You know, the top two are going to be both connected to stearate. So that's easy enough because it's you've got your ester linkage, and then this carbon starts an 18 carbon chain. So it's this 16 more carbons and then CH3 at the end. Now down here, it gets a little more interesting, but what always stays the same is you're always going to have this bonded to a phosphate. So this alcohol group here is going to become, you know, an oxygen that's bonded to a phosphorus. That's then bonded to another oxygen. This phosphorus has to be bonded to 4 total oxygens here. Hence, it's a phosphate. And then it was bonded to, what did we say, serine? Serine. So serine is this one here. So this phosphate is going to basically bond to this. So you get the phosphorus bonded to an oxygen, and that oxygen is going to take the place of this alcohol. So that alcohol goes away. We then are bonded to this carbon, this carbon, this carbon, and just draw this structure exactly like it. So going back to the answer here, this is again serine down here. That's our amino alcohol for this phospholipid. We have phosphate here, and then we have our CH2 over here. So this is the trickiest part down here. This is just the same as our triacylglycerol stuff from before. This is the new part here. So again, this carbon bonded to an oxygen, that's going to be bonded to a phosphorus. Phosphorus is bonded to 4 total oxygens, right? And then on the right side, that's where it's going to be bonded to the amino alcohol. Okay, so what's even trickier than glycerophospholipids is sphingomyelin. So the amine group of sphingosine is going to form an amide bond to fatty acid rather than an ester bond. The hydroxyl group will actually form an ester bond with phosphate, and that's going to continue on to a phosphoester bond, just like what we were seeing here for the glycerophospholipids. Oops, yeah. So here's an example of sphingomyelin. Now, you don't have to memorize the structure of sphingomyelin. You just need to be able to recognize it based on the condensed structural formula that we have here. So here's our condensed structural formula. You form an amide bond with the fatty acid here, and then a phosphoester bond that's going to connect the phosphate group to the glycerol, or I'm sorry, to the sphingosine, to the amino alcohol. Okay, so again, I'm not going to ask you to be drawing these sphingomyelins on the test. I just need you to be able to recognize them based on the structure that's drawn here. Here's your sphingosine backbone that you can use to identify it. Okay, so here is, for example, you might see this on the homework. If you're asked to recognize draw a sphingomyelin that has a particular fatty acid and an amino alcohol, all it is basically is it's the same backbone structure here for sphingosine, then an amide linkage to whichever fatty acid it is. So in this case, we were asking for palmitic acid, and then the amino alcohol choline. So this backbone stays the same, where you have this weird looking sphingosine here. Then there's an amide linkage from this nitrogen to the fatty acid that you're given, and then the same phosphoester bond that we saw in the glycerophospholipids to the amino alcohol. Okay, so again, you just need to be able to recognize these for the test. But that does it for today's lecture. We're going to carry on, and we're going to talk about steroids next. And then we'll finish up the chapter by talking about cell membranes. Until then, I'll talk to you later, and bye-bye.